Okay, good morning, everyone, and on this gorgeous, gorgeous spring day. I hope that everybody is beginning to be able to feel like they can start to move around outside again and start tentatively socializing. I urge you all, please, to get to vaccines as soon as possible. Extremely important that as many people get vaccinated as quickly as possible. If you have questions about the COVID-19 virus or the COVID-19 pandemic, and especially if you have questions about vaccination, please address them to me. Send me an email and ask me those questions, and I will try to answer them in class. Okay, it's a good opportunity for us to increase our knowledge base. All right, so let's get started. Try moving through our discussion about the nervous system, about nerves and the nervous system. This is where we ended off last time, and I'm described here what happens at the axon terminal. Remember, we talked extensively about how we generate an action potential, an electrical signal that runs through the axon of the nerve, but excepting in very specialized circumstances. That action potential comes to the end here and is unable to jump across this little gap here, which lies between the axon terminal there and the succeeding cell, or the postsynaptic cell. This is referred to as the synapse. It's where one cell abuts on the other, and somehow or other the message has to be conveyed across the synapse to the succeeding cell. This cell that lies on the other side of that synapse, the postsynaptic cell, can be of many different kinds. It may be another nerve cell in which we need to generate a nerve impulse. It can also be a gland which is going to be stimulated by the action potential, or it could be a muscle cell which is being told to contract. In all of these instances, the action of the cell is determined by the nature of the receptors which it has in its cell membrane. So we are going to take as our major example what are referred to as ligand-gated receptors. And these are receptors, these are channels, ion channels, which open in response to the binding of a particular substance, a ligand. Ligand is a substance which binds to a receptor. The ligand in this case is a neurotransmitter. Neurotransmitters are manufactured by the nerve cells, and they are stored at the axon terminal. They're stored in vesicles, and those vesicles are only expressed when an action potential arrives. So that's the sequence of events. An action potential arrives, and the vesicles containing the neurotransmitter excrete the neurotransmitter into this cleft. It diffuses across the cleft. This happens very quickly. We're talking about a microscopic distance, so it happens really quickly. The ligand binds to the receptor on the cell membranes, and in this case, what is happening? We are allowing the inflow of sodium into this succeeding cell and the outflow of some potassium. What is that going to do? It's going to depolarize this part of the inside of the nerve. It's going to depolarize that membrane. And if enough of such stimulation, if enough of these open, and especially, as we'll see in a minute, if there are enough of these terminals doing it all at the same time, then a new action potential can be generated by the depolarization of the membrane. So that is in the classic kind of story for how a nerve impulse is propagated across the synaptic cleft. Just think for 1 minute, though, about some of the other instances that I've told you. Think in particular about what if this here is not another nerve cell where nerve action potential is generated? What if this is a muscle cell? How is muscle cell actually stimulated to contract? Well, what controls that, again, is the fact that the muscle cell has in its cell membrane receptors, but these are receptors not to the kind of neurotransmitters that would generate a nerve impulse, but instead to a neurotransmitter called acetylcholine. A nerve impacting on a muscle and generating a muscle contraction releases acetylcholine, which is picked up by the cell membrane of the muscle cells, and that results in the muscle contraction. The take-home message is that we're going to find that there are many different kinds of neurotransmitters. There are many different kinds of neurotransmitter receptors, and this gives a tremendous diversity to the results of the arrival of an action potential. It all depends, when the action potential arrives, it depends on what it is impacting on, the type of cell it's impacting on. It depends on the type of neurotransmitter that is being released. And it depends very strongly on the type of receptors that are present as well. Okay, so let me just see. The first, there's a little complication now. I've talked always, we have so far talked always about a nerve impulse, for instance, arriving at a synapse and generating a new nerve impulse or generating some kind of positive action. In actual fact, there are also nerves, many of them, which are inhibitory. And any one nerve may have impacting on it many different axon terminals, some from inhibitory and some from excitatory cells. So this sounds rather strange. Why? What does an inhibitory synapse do? It suppresses. It actually hyperpolarizes the succeeding cell. The excitatory synapses, what they do is they're the ones that stimulate. They stimulate a new nerve impulse or whatever. The inhibitory ones actually stop it from happening. Why? Why do that? The answer is, of course, control. It's control, and it also allows a nerve cell, a cell body, to integrate information. It can integrate information from many different sources. And let me just show you quickly here. Here is a cell body with nerve endings, with axon terminals impacting on the cell body itself. These are the thousands and thousands of synapses. And these synapses, of course, we can't tell just looking at them like this, but these synapses may be from a mixture of excitatory and inhibitory synapses. So this cell, it responds to a summation of input. There may be many, let's say there's a lot of excitatory input and a little bit of inhibitory input. It would still generate a new action potential or response. Same idea. If there was a lot of inhibitory input, then it would result in the suppression of the nerve impulse or the action potential of whatever sort it is. So you should begin to get the idea now that that nerves can actually interact in all of these different kinds of ways. They can be excitatory, they can be inhibitory, they can be, it can depend on what neurotransmitter they're releasing, et cetera, et cetera, et cetera. In all of these complex interactions imply processing. processing, and also that we can begin to think that the nervous system may begin to be able to store information by this kind of integration. Let me just check that I've got everything. I've said everything that I want to say. Yes, on the issue of the neurotransmitters, I said that the neurotransmitters are released at the axon terminal and diffuse across. Well, there are actually various fates that the neurotransmitter can enjoy. First of all, when it's released, it needs to really reach a destination to start an outcome. But you'll realize that that neurotransmitter doesn't actually diffuse into the succeeding cell. Instead, it drifts across and it binds to a ligand. But it can also be released from that ligand. And very frequently, in most of our synapses, the synapse can actually recycle its neurotransmitter. Now, this is important because you just want the neurotransmitter to generate a single signal. You don't want to generate neurotransmitter and then have it hanging around for ages and ages and ages, regenerating a signal over and over again. Instead, the cell rapidly can take up the neurotransmitter again, and it can be recycled. Sometimes it's taken up by surrounding cells as well. And in some synapses, there is an enzymatic degradation of neurotransmitter. But for the most part, cells recycle their neurotransmitter. So here is the origin of a set of therapeutic measures which impact on nerves. And this is in the treatment of depression. You probably have heard of drugs such as Prozac. These sorts of drugs, they are referred to as selective serotonin reuptake inhibitors. Just think what I've just said. Selective serotonin reuptake inhibitors. Serotonin is a very important neurotransmitter. It has many different functions in the nervous system, depending on what nerves it is impacting on. It appears that in cases of depression, serotonin levels are low. So what a drug like Prozac does is it stops the cell, it stops the axon terminal from rapidly recycling the serotonin that it releases. That's why selective serotonin reuptake inhibitor. Prozac stops the cell from taking up the serotonin which has been released in response to an action potential. It allows the serotonin to hang around in the synapse longer, and it allows the serotonin to act for longer so that it produces a stronger signal in the succeeding neurons. And the impact of that appears to be that one of the impacts is it relieves depression. It increases the activity of nerves. It's believed to be especially in the cerebral cortex, where emotion and everything lie. Okay, so just keep this picture in mind. Underneath here, there's a cell body impacting on it. Many, many, many different axon terminals from many different nerves of different kinds. This cell body is in itself acting as an integration center. Okay, I'm just not going to discuss it in enormous detail, but just to tell you that the study of neurotransmitters is an area of enormously active research. There are many, many different kinds of neurotransmitters which are released by axon terminals. And not only are there lots of different kinds of neurotransmitter, but there are many kinds of receptors for those neurotransmitters. And those, depending on what receptor the neurotransmitter impacts on, you might get a completely different outcome. And so there's a huge amount of diversity in the action of neurotransmitters and of their receptors, depending on what cells are interacting with one another. So here's just a short list. I'm not, I'll tell you the ones I want you to remember, and really I just want you to remember those with a name and a basic function. And the first one we've already mentioned, acetylcholine. Acetylcholine is a neurotransmitter which is released from motor nerves primarily, and it causes contraction of muscle. But it has also been implicated in really diverse functions like memory formation and learning as well. This group here, they're called the biogenic amines. You don't need to remember that. But these you may have heard of. They include neurotransmitters such as epinephrine and norepinephrine. You'll remember those because they were produced by the adrenal gland, and those are the fight or flight neurotransmitters that produce all of the symptoms of alarm and of the need to run or to fight. But these are also strongly implicated in day-to-day awareness as well. So these are hormones, neurotransmitters. Yes, they are hormones as well, but we heard about them as hormones before, but they are actually also neurotransmitters. The others, dopamine and serotonin, I would mention those because you will very, very frequently hear them referred to as they're commonly talked about in terms of being involved with pleasure and things like this. That is correct in a way. They certainly are, but dopamine and serotonin occur very widely through the nervous system and have many, many, many different functions. It all depends on what nerve is releasing them, what nerve is picking them up, what receptor is binding them, etc., etc., etc. Serotonin, I've already mentioned as being implicated. Low levels of serotonin activity are implicated in depression, but imbalances in serotonin and dopamine in the brain are implicated in many different disease situations as well, such as schizophrenia. So it's obvious the dopamine and serotonin have, along with many other responsibilities, they appear to be very active in personality and development of personality and a sense of how do we say it, of appropriate interaction with the environment. There are also some neuropeptides which act as neurotransmitters. One of them that you may have heard of are the endorphins. And endorphins are neurotransmitters which are released under prolonged stress and pain. They are endorphin means a natural morphine product. And what they mean by that is that they have an activity which is very similar to a morphine product. They dampen pain principally, okay, endorphins. And those of you who are runners or athletes, you've probably heard of an endorphin rush that people get when they have stressed their bodies to a certain level. All of a sudden, they get a feeling of regeneration as endorphins begin to surge through the body. Another interesting one is this, a gas, nitric oxide, NO, is strongly implicated in contraction of smooth muscle and is also implicated in dilation of blood vessels. And for that reason, it has come under particular scrutiny for its role in sexual arousal. And in fact, drugs like Viagra act on the receptors for nitric oxide and enhance its ability. That's how they work. Now, these ones here, glutamate, gamma aminobutyric acid, which is commonly referred to as GABA, glycine, these we're not going to talk about in any great detail. If you want to, there is some discussion of them in your textbook. These are very strongly implicated in modeling of the nervous system, in restructuring function of nerves for the purpose, for example, of learning, of memory and learning, and those kind of functions. So these are very, very important systems, but it's a complex system, and it's beyond the scope of what we need to know. Okay, so this now moves on to our next chapter, and this is on nervous systems themselves, not so much the function or individual function of the nerve. Nervous systems are almost universal throughout the animal kingdom. There's only one group of animals that do not have some sort of intricate nervous system, and those are the sponges. And even in the sponges, there are cells which do respond to stimuli and which produce a response in the animal. They're just not true nerve cells. But once we move into the animal kingdom itself, even the simplest of the animals have some kind of nerve system. We're so used to that term, nerve system, that you don't really think about what it actually means. It means literally the system that we produce a system whereby our nerves interact with one another, and they interact for a very important purpose. And that is to produce a coordinated response. Just think about it. If we got sensory nerve cells all over our body, that's fine. You've got sensory nerves all over the body. What use is that to an animal if the information coming in is not somehow processed in order to produce a response? A nerve on its own is a wonderful thing, and it does all sorts of wonderful things, but it is pointless if it is unable to produce an appropriate response to a stimulation. And that appropriate response is produced by the organism being able to receive information, integrate the information, somehow or other, even in the most simple way, analyze it, and then have an output that produces a response to the information that is received. This can be in the form of a very simple system, but I should warn you that actually they must find that even the simplest systems are in fact very, very complicated in the way they operate. But if we look at the organisms like Hydra or things like that, which are radially symmetrical, we see there some of the simplest of the animal nervous systems. All of those radially symmetrical organisms, sea anemones, Hydra, jellies, those sorts of things that have a radial symmetry, have a huge disadvantage in actual fact, and that is that they receive information equally from all the way around them, and they need to integrate that, and they do so by having a nerve net. They have no brain system, they have no ganglia, or anything like that. Instead, the nervous system is relatively simple and is organized as a nerve net. There are higher organisms which also are radially symmetrical, and these are the sea stars, like the echinoderms, which are actually quite closely related to us, but they also have a radial symmetry, and it points to the huge disadvantage. A radially symmetrical animal does not have a front and a back. Instead, they receive information equally from all the way around, and they need to integrate that from all the way around. So in the sea stars, for example, the closest that they get to a true integrating system is to have a nerve ring which receives information from all of the arms. But that radial symmetry constrains the organism, and it especially constrains the development of its nervous system. So even in the sea stars, the nervous system remains rather simple. As soon as you go to a bilaterally symmetrical organism, we see something happen, which drives the evolution then of nervous systems throughout the animal kingdom. And that is that in the bilaterally symmetrical organisms, even in the primitive ones like the flatworms, the planaria, there is a front and there is a back. And that means that the front has the potential to be the major receiver of incoming information, incoming sensory information. Makes sense. If you have a front that is always going first, you want that front to be able to see what is happening in the environment. That leads to the development of extensive nerve systems in the head region. And much of the integration of that information is going to take place right there at the head, in order that the organism can make decisions, can send output to the rest of the body. Something bad happening up here, enter into avoidance, turn the head, turn around, go back, whatever. That processing, extensive processing, starts to take place right here at the head and leads to the development of an extensive associations of nerves, which we see physically as in large collections of nerves interacting with one another in a brain. So even in the very primitive bilaterally symmetrical organisms, there is a brain. And that is a tremendously important evolutionary development, and one which now then becomes, we see, exploited extensively through all of the rest of the animal kingdom. Look at things like the insects, and you see the same kind of thing. They have a head, extensive sensory apparatus at the head. That's where they have got a lot of, not all, but they are able to smell, see, taste, etc., all at the head, and all of it is getting processed here in the brain, and then information is sent out. You probably know this can reach really high levels of development in mollusks such as the cephalopods, the squid, octopus, these sorts of things. There is a quite large brain which is capable of extensive analysis and extensive amounts of learning. Even though these organisms are often short-lived, nonetheless, I hesitate to use the word intelligence without providing a definition of what intelligence is. But these organisms are extensively able to analyze situations and produce a complex output from their brains. In the vertebrates, the same kind of idea occurs. We have a brain up in the head, which, if we move through all the vertebrates, we realize the brain gets more and more and more complex until in the really higher vertebrates, the brain can be extremely large and capable of extensive analysis, extensive learning, and apparently extensive intelligence. I want to just point out a little bit some of the differences, though. I want to remind you that in these groups here, in things like planaria, the insects, and in the mollusks, the nervous system is a little bit differently organized to the way that it is in the vertebrates. And in that, although there is a brain, there is a nerve, the nerve cords. The nerve cords are the communication system from the brain to the rest of the body. And in these groups, that nerve cord is ventral. That is, it runs down the tummy side of the organism. And it is very often based on a double nerve cord system. The nerve cord looks like a ladder. And there can be elaborations on that, but that is the basic form. But when we come to the vertebrates, then in the vertebrates instead, our nervous system is based on a system of a tube. We have a hollow nerve tube, which is dorsal. That is, it runs down the back. And that is true for all of the vertebrates. The nerve cord is hollow. Even our spinal cord, if we section our spinal cord and look at it, we'll see that there is a hollow that runs through it. If we look up the brain here, we'll see that the brain, in fact, is an elaboration of a tube. And we'll see that again in a minute when we just refer briefly to development. So this is the human brain. A couple of things to point out to you. Well, it's very large. Some vertebrates have enormous brains. The size of the brain is not a reflection, however, of the amount of intelligence or whatever. A very large animal has a very large brain because it needs a huge amount of motor output to manage that giant body. Because a whale has a huge brain doesn't mean it to say that it's hugely intelligent. Those functions of intelligence, the ability to extensively learn, et cetera, appear to be functions of the very outside of the brain, of the cortex of the brain. And we'll see some more of its structure in a while.
Professor Muir?
Yes, go ahead.
I just wanted to know if intelligence is hereditary.
You know, it's such a fraught subject. First of all, I'll just say this. Our ability to measure intelligence is very poor. I think our ways of measuring, it is true. It does seem that on the tests that we use for IQ and everything, that there is an association, that there is a genetic association. But having said that, I'll also say that there are outstanding exceptions. There are many people, you know, who are plainly and obviously extremely brilliant and intelligent, everything, whose parents appear to just be, you know, of ordinary intelligence. And the reason it gets so complicated is that there are probably many genes which interact to result in what we perceive as being intelligence. And so a person is subject to a lot of influence as they're growing up and everything else from all sorts of things, probably other than just their genes that they inherited. And I know that sounds like a very vague answer. It's not a specific answer to your question. There are certainly many aspects of intelligence which appear to be inherited, genetically inherited.
So do you think it's more innate?
I think your genes provide you with the basic building blocks. But I would say that your nervous system is very plastic, and that my personal belief is that in fact people can develop intelligence as well.
Okay, I get you.
I understand. Inherit. Yeah, like
okay.
you inherit a basic building blocks, and what you do with the building blocks is.
That's true. Yeah, I think so too.
Yeah.
Okay, that makes sense.
Okay, so here it is. This is our brain. I'll just point out a couple of things to you. This brain is pointing that way. It's looking forward. You can always tell the orientation of the brain by looking at a couple of bits of its anatomy. This body here, there, is called the cerebellum. And it's at the back. It's behind here. You see this here, that lobe there, that is called the temporal lobe, and it always points forward. So this is the front of the brain here, and it's in this area here, by the way, that we believe all our sense of personality, consciousness, and everything else, that's where that kind of processing takes place, is at the front of the brain here. At the back here, you can see this is the spinal cord going off down the spine and communicating then with the rest of the body. So what you're looking at there is dissected out a portion of the nervous system called the central nervous system. And that is the brain and then the spinal cord. All of the other nerves, all of the nerves which are running through the rest of the body, are called the peripheral nervous system. And the peripheral nervous system is this extensive set of nerves, which are, first of all, sending information to the brain, but then the brain is sending out instruction to the rest of the body as well, also through the peripheral nervous system. There's one exception to this. There is one portion of the nervous system which is really important, but which does not depend upon any processing through the central nervous system. And these are the reflex systems. And you all know, you go to the doctor, they tap your knee to test your reflexes. Well, this is a real thing. When they do that test, what happens is the doctor actually hits a tendon in your knee, and that provokes a stretching of your quadriceps muscle. Well, when you, in just ordinary activities like walking and that sort of thing, your muscles continually measure the amount of stretch that they are under as your muscles operate. And this is actually to provoke a coordination with the the opposing muscles so that you can walk properly, etc., etc. If the doctor taps your tendon like this, the doctor induces a sudden stretch of this muscle, very, very small. The response of your body is actually quite profound. And the first thing you realize, of course, your leg kicks up like this. And the reason it kicks up is because this muscle actually contracts. It's received a piece of information. It's been stretched. The response is to contract, to get it back to its original shape. And that causes your leg to kick out. Here's how it works, though. There's no processing by the brain. Instead, this is a simple processing system which runs right through the spinal cord and it never reaches the brain. When that little receptor in the muscle is stretched, it sends a message through this system, which is called a reflex arc. And it communicates directly with a motor neuron, which runs to the muscle. So you pull the stretch this little stretch receptor, it immediately sends a message to the muscle and tells the muscle contract. There's no processing by the brain, and it happens almost instantaneously as soon as the message can make its way around. But it's a little bit more complicated than just that, sending a message. In actual fact, the reflex arc also contains a segment which is inhibitory. Remember, I told you before, neurons can be either stimulatory or they can be inhibitory. And this reflex system contains a little inhibitory neuron. And what that is doing is it's inhibiting the contraction of the hamstring muscle. temporarily, the hamstring muscle is unable to contract. So the only muscle that can contract is this one, is the leg, and your leg kicks up. This is important simply because this is one of the simplest processing systems that you can visualize. But you see that in response to this stimulation, the nervous system has, in a way, analyzed it and has resulted in an output. The output being twofold. One, stimulatory, contract that muscle. The other, inhibitory, relax that muscle, keep that muscle relaxed while this one's contracting. Okay, most of the other processing that takes place is extremely complex and depends upon the central nervous system, the involvement of the central nervous system. So again, we are vertebrates and we have a dorsal hollow nerve cord. And this, even when it reaches the brain is in fact an elaboration of that tube system. And the hollow inside that tube becomes quite extensive in the brain itself. And it forms these hollows here called the ventricles. So here, running up the spinal cord is the central canal. When it reaches the brain, the central canal kind of opens up into these cavities inside the brain. You don't need to remember all the names. Just be aware of the fact that they are quite elaborate in structure. They have very important functions. The first is that they are filled with a fluid called cerebrospinal fluid, which is derived from the blood and is rich in nutrients, rich in oxygen, and is kept in circulation through the cerebral ventricles because the walls of the ventricles are ciliated. So this fluid is in continual motion. The amount of fluid in there is very carefully regulated. There are receptors which determine the volume and the pressure and maintain a homeostatic control. There's something important, though, that is not shown here on this diagram. And that is that the brain and the spinal cord are surrounded by membranes. There are, in fact, 3 membranes which surround them. And between 2 of them, there is a layer of fluid, and that is also cerebrospinal fluid. And it communicates with the cerebral ventricles. There's actually ducts which connect the cerebral ventricles and the fluid surrounding the brain. The fluid surrounding the brain effectively floats in that fluid. It's cushioning, but it's also nourishing. It's in continual movement, and it's continually renewed and provides nutrition and oxygen and everything to the brain, in addition to everything that's coming in through the blood system. So you don't need to, as I say, you don't need to remember the names of the different ventricles. Just be aware that there's this extensive ventricular system. So that's the central nervous system, the brain, the spinal cord, but then all of the nerves which emanate from that and which go to and from the central nervous system, we refer to as the peripheral nervous system. I will just point out to you quickly, there are actually kind of 2 large sections of the peripheral nervous system. The one that runs through the body and everything, which is easy to visualize. But then there are a set of nerves which emerge straight from the brain. They do not emerge from the spinal cord. Most of the peripheral nerves emerge from the spinal cord. The exception of these, which are called the cranial nerves, and they emerge directly from the brain. There is something that this diagram is not 100% accurate, because this shows that these cranial nerves as only being here at the face. It's true that many of the nerves of the face are cranial nerves. They're derived straight from the brain, not from the spinal cord. But the inaccuracy is that there are nerves which run all the way down into the body as well. In fact, one of the most important of all of our regulatory nerves, regulating breathing, regulating blood pressure, all sorts of things, is the vagus nerve. And that is a cranial nerve, which runs all the way from the brain, all the way down into the body itself. Still part of the peripheral nervous system. It's just a different section of the peripheral nervous system. Okay, so the brain is organized. Most of the executive function of the brain, if you like, takes place in a thin outer layer here, which is the cortex of the brain. And the cortex consists of gray, it's called gray matter, and literally does, when you look at a section of a brain, it does indeed have a kind of a gray look to it. And this is a collection of nerve cell bodies, extensive dendrites, and also, very importantly, interneurons. I showed you that an interneuron in the reflex, in that reflex arc. An interneuron is a neuron which runs between two other neurons, and which does not involve any involvement of the brain or anything else like that. Interneurons are connecting neurons, and they become very, very important in integration of information because they may be excitatory or they may be inhibitory. So they can modulate the information which is being put into another neuron. So that's gray matter here, largely composed of cell bodies, dendrites, interneurons, all of these things which we now begin to understand are involved with integration and analysis of information. A lot of the body of the central nervous system is made up of white matter, which is largely nerve axons, which are myelinated. They have that fatty lipid myelin sheath around them, which is the insulator of the axon. Here in the center, you can see the ventricles here. Now, in addition to that, there is a large number of cells which are called the glial cells, or the glia. And I see here this statement here, the glia are specialized macrophages that support neurons. Not all the glia are specialized macrophages, some of them are. So that is partially correct and partially incorrect. But the glia are cells which support nerve cells. They appear to nourish cells and kind of look after the nerve cells, but which also seem to drive their development. And when new neurons are produced, for example, we believe that it is glial cells that may guide the direction in which new nerve cells grow and guide the associations that they make with other nerve cells. So the glia are subjects of an enormous amount of research. They have many different functions inside the nervous system. So these are the three kinds that we'll talk about. There are others. The first are called the astrocytes. Actually, I think, let me go here. Okay, these are the astrocytes here. Astrocytes, they're shaped like a star. And the astrocytes have many different functions. They've been shown to have, but one of the really important functions of the astrocytes is they become associated with all of the blood vessels in the central nervous system. And they induce the cells of the blood vessels to develop what's called tight junctions. The boundaries between the cells are very tightly bound with specialized proteins that stop any leakage of material between cells. In the rest of the body, those cell boundaries are weak areas, which are actually exploited as kind of channels for movement of substances from one area of the body to the other. In the brain, though the blood cells, in particular of the blood vessels, are very tightly bound to one another. And it is very difficult for most substances to make their way from the blood system into the brain itself. And this is called the blood-brain barrier. And the blood-brain barrier prevents many substances from making their way into the brain. It does not stop nutrients, it does not stop oxygen, all those kinds of things, but it will stop toxins or excretory byproducts, those sorts of things. And in addition, it will stop many, many drugs, many therapeutic drugs cannot make their way into brain tissue across this blood-brain barrier. That's all maintained by the astrocytes. Another set of cells, especially important in the brain and the spine itself, are the oligodendrocytes. This is another group, and they drive the formation of the myelin sheath around nerves. Then the last set here are almost the most interesting of all. These are called the microglial cells. And these are the cells which are modified macrophages. They're actually blood cells. They originate as blood cells. They move into the central nervous system, and they have many different functions. They do the normal kind of macrophage things. They eat dead cells, they eat damaged cells, they clean up excess protein, all of these kinds of housekeeping functions. But it's also been shown now that they are important, for example, in memory, in formation of memory, and that disorders of memory are sometimes associated with disorders of the function of the microglial cells. And it may be, this is just the kind of theory, it may be that microglial cells actually guide the formation of new neural connections, which are necessary to form in the formation of memory. So a very interesting group. We won't talk about them anymore, but the glial cells are a subject of an enormous amount of research at the moment. I'll just point out one thing to you. There is in this diagram, the wall here of the ventricle, also with tight junctions to it. But here's the ventricle, and you can see that these cells that make up the wall of the ventricle are actually ciliated. Keep that cerebrospinal fluid in continual motion inside the brain and the spinal cord. Okay, so let's just finish off with this, which will introduce us to our next topic. This is just a diagrammatic layout of the whole of the nervous system. So you think about it in the way in which the nervous system functions. First of all, there is a huge amount of sensory input, which needs to be somehow processed. So that's got to get to the brain in order to be processed. So all the nerves from the peripheral nervous system feeding into the central nervous system, these are called the afferent neurons. Afferent meaning towards, and in this case, towards the central nervous system. So there's all of the sensory input is sent to the central nervous system. The central nervous system actually analyzes it. It actually places it and organizes a response. That response is by motor output. So the motor output goes via efferent neurons. But that output is not necessarily to effect a movement, a motor movement, a voluntary movement. It may. The output may require some form of control of muscle movement. In other words, it's going to affect a physical behavior in response to the sensory input. But a huge amount of the output of the central nervous system is actually not under voluntary control at all. is through a system of the nerve section of the nervous system called the autonomic nervous system, the unconscious nervous system. And this has three divisions, two major, one we'll regard as being relatively minor. The two major divisions are the sympathetic division, which you can think of in broad terms as being the excitory. It's a sympathetic nervous system when you get a fright that drives you into your fight or flight mode. The parasympathetic nervous system in general, not entirely, but in general, is the calming part of the autonomic nervous system. These two operating in opposition to one another to effect fine control of the state of arousal of the organism. The third unconscious autonomic part of the nervous system is called the Enteric Division. And this is purely associated with the gut and with organs of the gut, like the pancreas, gallbladder, that sort of thing. And it governs actions of the gut, glandular release, and peristalsis, all of these sort of things. But in fact, the sympathetic and parasympathetic nervous systems exert strong control over the enteric as well. It does have its own independent activities, but those activities can come under the control of the sympathetic and parasympathetic as well. So we'll leave it there for today, and we'll complete this at our next class. Yes. If you have any questions.
I have one question. Starting on Monday, does this chapter cover like the parts of the brain?
I beg your pardon, does what cover that?
This chapter, does it cover like the parts of the brain?
Yes.
And we'll start that like on Monday, right?
Yes.
Okay.
Yes. What do you have a particular interest?
I do have a little interest in the parts of the brain. That's why I asked.
Okay, good. Okay, well, be sure to, because, I have to do this like just in survey kind of mode. So be sure to ask your questions, and I hope I can answer them. But be sure to have your questions ready to ask on Monday.
Okay, enjoy your weekend, Professor.
You too.
Hey, Professor.
Yes, oh, hello. Where are
Hi.
you?
I'm right here.
Oh, there you are, yes.
Question. So I was reviewing the email that Pearson sent me because we were discussing
Yes.
MyLab mastering. It's saying that I need to buy the textbook again because my free access code is expired.
But didn't you buy it originally?
No, because I believe everybody got it for free in the beginning of the year.
No. Yeah, each semester you have to buy access.
Okay, so
Yeah, I'm sorry that wasn't clear.
No, that's perfectly fine. I'm just confused. So
Yes, I know.
I'll buy the
You're not the only one.
Okay, no problem. When I get paid, I'll buy the textbook and then I'll do the assignments.
All right. Okay. Okay, Nicholas. I'll bear that in mind.
Okay.
You are not. Quite apart from me being confused, there are other students who've run into the same problem.
Oh, okay, no problem. I just wanted to clear everything up.
Yeah, that's okay. That's fine.
Okay, no problem. I'll see you.
All right then. See you on Monday.
See you on Monday. Bye bye.